Alright, ladies and gentlemen, here we are. We're going to do a quick little video for getting you guys started on this lab, viral DNA integration. So this is found, let me show you where this is at. So you're in transcription. We're specifically looking at how viruses carry this out. Okay, so we're going to be looking at this. We felt like it was kind of a good way to go since we're all stuck at home because of the coronavirus. So anyways, if you like my background, you guys, I'm trying to make you feel like you're in a good little world. Look at that. See? Can't even hear the streams, but trust me, there's a stream behind me. Okay? Here, I need to move this around because I keep looking at it, and then I feel like I'm looking at myself instead of you guys. So here we go. All right, so you guys, what we're going to be doing is, again, we're going to be doing, we're working on this one right here, looking at how does a virus carry out transcription. What's interesting about viruses, as you guys know, viruses cannot do this by themselves, and so that is why they require our cells to do this. And so viruses will attack our bodies, our cells, to carry out transcription, and then a later process that you guys will learn about called translation, to make the actual protein to make itself. And so we're going to be really looking at this first part here. Now to do this, it's going to be a little hands-on for you guys. You'll need some material. I'm giving you guys multiple options though, and so hopefully you have the material for this. If worse comes to worse, you can just draw it and we'll probably count it as that, okay? But if you have some of this material, I mean, all of you should have paper, so you guys can do this. So when you're doing this, you guys, please read the instructions, the introduction here. I'm not going to go over all of this. I just want to go over the key parts to it. But what you guys will, when you read this, it's going to go over a couple key things. The first thing is that we're going to be looking at specifically HIV, which is a retrovirus, is what it's called. And HIV is, if you guys don't know, it's the virus that causes AIDS. So it's a sexually transmitted disease. And there's a lot of people and populations in the world that do have a high amount of HIV. And when you have that, it doesn't always have to pass on sexually. It could just be in bodily fluids. It's where it's passed on. So some people get HIV infection and did not, and it's not like they're sexually active. They're born with it sometimes. And so there's some countries that this is a pretty big deal. And to look at this and see how it kind of works, we're going to kind of dive into this. So the first thing you guys will want to do, and I'm not going to go over this, so I will want you to kind of pause the video if needed. If you need to, you can even open up this video in a separate tab, and then you can keep working on this. But anyways, you'll go to this thing right here. Okay, so here I'll open it up for you so you can see what it looks like. It's not going to work, is it? So I'm going to have to copy and paste this puppy. Control C. Make sure, okay. Then when I pull this up, Control V, it'll bring up this little activity or a little video that you guys can watch. Kind of we'll go through what's going on here. After you watch that, you'll then go through this little click and learn. So you have to do the same thing here. And I might try and fix these links so that you guys just have to click on them, but until then, you're going to have to deal with it. Alright, so here you go, and then you can just launch the interactive. These should work on your iPads. Anyways, we're going to click on HIV. You can actually look at some other ones, which is kind of interesting. Do they have coronavirus on there? Let me go back. They don't. Okay. Anyways, so you can click on HIV, and it'll actually go through some different things. You can look at some different structures here. Yeah. So very cool. Okay, and so using that, let me go back to my document. It'll have you look at some different things. So it says draw a diagram, explain the basic structure of HIV, and you'll do that from that this virus explorer thing that I just pulled closed right here. Okay, and so you can pull this thing up just like so, and you can look at relative sizes of these things. As you guys can see, the HIV virus right there, it's rather large, but look how big the Ebola virus is. Kind of huge. Then the Zika virus, which was just a couple years ago. Kind of crazy. Influenza A, this is kind of like your flu stuff. It's a very powerful dangerous one, though. All right. So, anyways, you'll look at that there. You'll answer these. So, I'm going to let you pause the video. I want you guys to work on 1 through 5 really quickly by looking at those. Okay? So, you can pause the video there and work on that. All right, so I assume you guys are done answering these questions. I'm not going to go over these. I will read what you guys have there, and I'll give you feedback if you need to fix those. Okay, so now we're going to get into this one part that I want to kind of model for you. So right here, what we're actually going to do, you can do this with piece of paper, with different colors paper. You need 3 different colors of paper. You could do this with yarn. I'm actually going to use pipe cleaners for this. Okay, and I just have 3 different colors. So I've got blue, I've got green, and yellow. Okay, it's just what I had lying around at the house. And so what I'm going to do, I'm going to have my blue represent my DNA found in the humans in eukaryotic cells. And I've got 2 strands of this because I know that, I know the DNA. Now you guys know that I'm really not in a world like this, right? Yeah. Anyways, I'm going to have these 2 strands be my human, my DNA found in the human, in the host cell. Okay, and then I'm going to have green right here be my DNA, and I need to cut it. 1 second. I forgot I need some scissors. Let me pause this. All right, I'm back. Let me pull this back up there. Okay, so I've got my green. It's like blue on your guys' screen. Interesting. Okay, so anyways, this is green. Okay, and what I'm going to do is I'm going to just cut that. This is my viral DNA, okay? Don't tell my wife I just cut this with her scissors. Okay, so this is my viral DNA, kind of small. It's just kind of a short thing. And then I've got this right here that is going to be the DNA that gets transcribed, and I'm going to make it the same length, and it's yellow, as my viral DNA, except I'm going to make 2 of those. All right? So as such. Like I said, don't tell my wife. Okay, so I've got all the pieces that I need, okay, right here. And so just a refresher of what I've got 2 long pieces. This is my host DNA, or the cell, the DNA found in a human, or a section of it, right? I've got a green one right here that looks blue to you guys, but it's green, and it's kind of smaller, and I've just got 1 strand because viral DNA is an RNA, retrovirus, and it is single-stranded. And then this right here will be the DNA that will come from the retrovirus's DNA and will get integrated into the host DNA. Okay, so here we go. All right, so it says you will now make a model of the host cell genome and virus genome using the yarn and piece tape, whatever you guys want to use. Like I said, I'm using this right here. And use the longer strand to represent the DNA of the host cell. Draw a diagram of what you constructed model 1. Okay, so right here you can actually draw a picture of what this will look like here. Let me, and so anyways, I want you guys, you can either draw a picture, you can take a picture of it and embed it into this right here. Okay, whatever you want to do there. But ultimately, this is what it would look like. So my host cell genome will be this. Okay, my 2 strands. I'm going to show that it's double-stranded because it's DNA. So I'd have my double strand, maybe it'll be a double helix, which it would. So I'm going to show it kind of twisted around each other like so. Okay? For the HIV genome, well, I've got my 1 strand, and actually it would be kind of like maybe a single-stranded, kind of like that maybe. And so I could draw that there. And then my copy of the HIV genome would be this. So I've got these 2 strands. It's now DNA. Okay, and so it's going to be like that. Okay, so right down here you can write down some characteristics. Now this is just a review of what we've kind of already gone over in class before, in the module before. So write down some key characteristics of the host cell versus this versus this. When you're done with that, though, now it's going to have you guys answer a few questions. It says you're going to actually insert this into the genome itself. And then I want you guys to put a picture there. Now, just to get you guys thinking, there's so many places here's my host cell's genome. It's got to embed this now newly formed one that was created from this process of transcription. It actually does what's called reverse transcription. From the video, we'll talk about that. So here's this RNA. It's got to take the RNA that we would normally make in transcription to, we know that our cells read this DNA strand and would make an RNA from it, an mRNA, messenger RNA, that would then leave and go and get transcribed or translated and made into a protein. But what viruses do is they already have an RNA. They need it to be embedded into this. They can't just put their RNA in this. So actually what they do is they have the cell of the virus take this and say, okay, from this RNA, let's make some DNA. So kind of reverse of what our cell would do. And so then they have this and they can actually embed it. And it can be embedded in the middle. Like if you want to, you guys could snip this and put this in the middle. You could put it at the end, at the top, wherever you guys want to put it. And just show me how a diagram showing how it kind of uses reverse transcription to go back into that DNA. Now the issue that occurs is now those cells have that gene inside of their cells, inside of their genome. And so every time they are transcribing to make protein, they start making this section now and start to replicate and make copies of that HIV virus. And when that occurs, it can then spread to other cells, and that is how you get that's the sickness and that disease. Okay, and that's the same for a flu and a cold. They might not follow reverse transcriptase transcription, but they could do other processes to express that DNA. Okay, but specifically for HIV, that's what it's kind of doing there to do that. So I want you guys to answer these few questions at the end, and then submit this, and I will give you guys some feedback on your work. All right, so a couple things. You'll need a picture right here of your actual model. You'll need pictures here of your model or drawings, and then make sure you answer these questions. All right, if you guys have any questions, let me know.